Hi again, everyone. For those who didn't join us for the first half, welcome to our final Knauss Brown Bag Seminar of the Year. This is the second talk of the hour. Emily Cira is a Ph.D. candidate at Texas A&M University, Corpus Christi, and a 2018 Texas Sea Grant Knauss Fellow with the EPA National Nutrient Criteria program. She has been working in the water quality field for the past decade, and for her dissertation, she is describing nutrient-phytoplankton, not complicated, nutrient-phytoplankton dynamics in Baffin Bay, Texas. So excited for your talk.
Thank you. Okay. So switching gears a little bit from the last talk, I'm going to talk to you about one part of my dissertation project. And this is specifically looking at brown tide in Baffin Bay, and we're getting that data through a volunteer monitoring program. So I'll show you some of the results we're getting, and then I'll explain a little bit more about the monitoring program to close the talk. So I thought I'd start with why you all might care about Baffin Bay. And since I assume many people here work with fish, it's because it's a world-renowned fishing spot. All of the Texas fishing magazines call it the jewel of the Texas coast. It's a very rural area, but it still is really important to the Texas coastal economy. You can see here, this is a Texas survey of catch per unit effort for black drum, and this is Baffin Bay, the one that lights up along the bottom. And yeah, great fishing for black drum and also trout in red drum. Why I'm interested in Baffin Bay is because it's home to Texas brown tide, Aureoumbra lagunensis. This isn't a toxic organism, but it's a disruptive species. When it blooms, you can see here on the bottom, it shades out seagrasses. It's also a poor food source, so it can have detrimental effects on the systems it blooms in. There was a notable bloom in Baffin Bay. I think it started in 1989 and it lasted almost a decade. I think it's still the longest standing HAB on record. But because it's been limited to the Texas coast for so long, there hasn't really been much research interest in understanding why this organism blooms. But within the past 5 years or so, it's also been found along the Florida coast in Indian River Lagoon. You may have heard about that. And then also here in Cuba. So people are starting to get more interested in understanding the dynamics of this organism and what causes it to bloom. So back to Baffin Bay, there has been some very visible signs of eutrophication in the system. As I mentioned, we've had brown tide outbreaks and associated die-offs of seagrasses. There's been hypoxia and associated fish kills. And there's also been some other symptoms along the food web. There was a die-off of these benthic clams. And then back in 2013, fishermen approached my university wondering why the fish they were catching had what they called jelly flesh. And they were pretty much just emaciated, starving fish. And that's actually what started the volunteer monitoring program. In addition to that, there's been a long-term decline in water quality. So these figures here are from a Texas report showing temperature and some nutrients since the 1950s in Baffin Bay. I've highlighted Baffin Bay in green. And you can see overall, Baffin Bay is getting warmer and nutrient concentrations, orthophosphate and then Keldol nitrogen are increasing. So if you have warmer waters, it increases metabolism and cell growth, more nutrients. It's not a big surprise that there's been increasing concentrations of chlorophyll in the system as well. And I've highlighted here the periods where we've noted brown tide blooms. So it seems that that increase in chlorophyll over the long term is being driven by these brown tide blooms. And so we think Baffin Bay is becoming a more eutrophic system, but it's noted here, number 36. So there's really not insufficient data. This is from a NOAA report a decade ago. We still don't really know much about the bay. We can't make any conclusions about it being eutrophic or not. So the existing data is still too limited to tell us what's causing this degradation, and specifically for me, what's driving these brown tide blooms. And so we started a volunteer monitoring program in 2013. The volunteers visit 9 sites in the bay, but here I'm just focusing on these 5. So they go to these sites monthly and collect a whole slew of water quality parameters, and it's still ongoing today, but I'm just going to be talking about the first 3 or 4 years of data. And so from that, I've kind of selected 3 main interesting points to share with you, and then I'll talk about the monitoring program at the end. So the first trend I want to point out is the spatial trend in brown tide abundance. So here along the bottom, are the 5 sampling sites in Baffin Bay. Really the only one you have to worry about is this orange one here, which we're calling BB2. And this figure over here shows brown tide abundances in Baffin Bay for all those sites over the 3-year period. And you can see the orange bar jumps out from the rest as having high brown tide abundances. And just to give you a reference of what high is, when brown tides are noted in systems, it doesn't even have to reach like 1 million cells per mil for it to be considered a bloom. So overall, abundances are pretty high along the system. So in addition to that, there's a temporal trend that we noted. This is average abundance across all those 5 sites. In the first 2 years of sampling, abundances were generally high and they exhibited the classic seasonal trend, being high in the summer and then dropping down in the winter. But here, between 2014 and 2015, the spring of 2015, it was increasing and then it's just crashed and it's remained low ever since. And so for the last trend I want to point out, I've now overlaid the points and mainly just keep your eye on the orange ones. You can see over here, the orange site, BB2, was the only site where brown tide abundances kind of rebounded after that initial crash in the spring of 2015. So first, I'll talk about the spatial trends. And for that, to get everyone on the same page, I just threw this in here. Spatial trends are related to dissolved organic nitrogen. And in case you're not aware, there are many different pools of nitrogen. Here I'm differentiating them between inorganic and organic. And so dissolved inorganic nitrogen is generally very labile. A lot of phytoplankton taxa have access to it, where organic nitrogen is more restricted. And we're finding many taxa can access it, but it tends to be maybe the more harmful species, or most harmful species can't access it. So nitrogen form, along with overall nitrogen concentration, affects the phytoplankton community composition. And brown tide is interesting because brown tide can utilize organic nitrogen, but cannot utilize nitrate, which is one of the forms of inorganic nitrogen. Okay, so dissolved organic nitrogen concentrations are exceptionally high in Baffin Bay. This figure on the left shows organic versus inorganic, and you can see that dissolved organic nitrogen is about an order of magnitude higher than inorganic in Baffin Bay. And even comparing it to other systems along the Texas coast and worldwide, this bullseye here is Baffin Bay, and this is Keldol nitrogen. But you can see it peaks in Baffin Bay in South Texas, and this happens to be where brown tide had been isolated pretty much until 2013. And recent work in other systems where a brown tide has been blooming have also noted this relationship between dissolved organic nitrogen and brown tide. So even within the bay, organic DON seems to be shaping the structure of the community spatially. So here again is the figure showing brown tide abundance, highest at BB2, and DON concentrations are also highest at BB2. So DON within Baffin Bay seems to be driving the spatial distribution. And to get at a more, that's definitely a relationship, but to maybe point at some more causal mechanisms, we conducted a series of bioassays where we took a water sample that had dominating brown tide abundance, and we spiked it with different forms of nitrogen. We have ammonium and nitrate, which were forms of DIN, and then urea, which was a form of DON. And you can see this is a 24-hour growth rate, and only ammonium and urea showed growth over those 24 hours. So for this community that was dominated by brown tide, DON was as stimulatory as DIN, even though DON concentrations are already very high in the bay. And nitrate was not stimulatory when the community was dominated by brown tide. So overall conclusions about brown tide spatial trends: Baffin Bay has high DON concentrations, and this is related to why brown tide abundances are so high within the bay. And this has been noted in other systems. And even within the bay, we can see that happening at a smaller scale. Okay, so moving on to the next aspect I want to talk to you about, which is the temporal trends. And here, as I mentioned, you see the seasonally in high abundance and then the crash in 2015. And so a little bit about Baffin Bay. It's a semi-arid climate. It's hypersaline, very long residence time, high evaporation rates, very little tidal influence, And I was not expecting this, but the 3 years of monitoring covered very climatic extremes. When we started sampling, Texas was in the middle of a drought, which is relatively common for South Texas. But then 2 years into it, it started raining and it pretty much never stopped. So that's why we see, we're looking here at salinity. Salinities are high and consistent, and then they crash, and they're slowly creeping back up. So we have the general low rain and high rain period. During the low rain period, again, you see the typical seasonal trend in high brown tide abundances. But during the high rain period, you see this quick drop when the rain started. And even as salinity is increasing, the average is still staying low for most of the sites. And we know this is a plot of brown tide abundances versus salinity combining all the sites for all the years. And you can see that, yes, more like higher salinity leads to higher brown tide abundances. And this again has been noted in experiments in Baffin Bay prior to this. And this could be for a couple reasons. First of all, brown tide is well adapted to grow in high salinities, so it's going to potentially outcompete other phytoplankton taxa. And it can also be due to some indirect reasons. High salinities, usually grazer abundance drops off. And in high salinities, brown tide, one of the reasons it can withstand at high salinities is excreting this like a saccharide ooze that deters grazing, and which becomes more intense during high salinities. And also high salinities are representative of slower flushing rates. Brown tide grows relatively slowly, so if it keeps raining and flushing rates are higher, brown tide won't be able to outcompete other phytoplankton taxa. And again, salinity is also related to factors like temperature that might affect evaporation and then also be related to salinity. So it's just important to note that. But brown tide is related to salinity, and again, many people thought prior to this that you had to have hypersalinity conditions for brown tide to bloom. So some general conclusions: brown tide related to salinity, and this helps explain seasonal trends, winter and summer, and also the interannual variability with the dry period and the rainy period we saw in our brown tide abundances. Okay, so the last part I want to talk to you about is kind of a combination of the previous two, and it's something I just think was really interesting that we found. And so that was this rebound in BB2 after the raining started. So for that, BB2 seems to be a hotspot for brown tide. It had the highest brown tide abundances, it had highest DON concentrations, and it was the only site where abundances rebounded after this high rain period. Why is that? Well, Texas is interesting in that there is this gradient of rainfall along the coast, and I cut it off in the middle, but Baffin Bay is right here, so it's falling within that gradient of rainfall. So this BB2 site is, first of all, receiving less rainfall than the other sites, or the watershed that feeds into that is getting less rainfall, and the watershed is smaller than the other tertiary bays of Baffin Bay. So it would be less susceptible to flushing. In addition, if you've been to South Texas, you know the winds there are very strong and very consistent. In the summer, they are generally coming from the south, and in the winter, they come from the north. So having those wind directions, it's pretty much cutting off water movement between Laguna Salada, where BB2 is, in the main Baffin Bay channel. And there's no tidal influence really in the system. So if nutrients trickle in to this area, they pretty much aren't going to get out for a very long time. And we see that it's poorly flushed looking here, hopefully. The green and blue dots are the other tertiary bays, and you can see during that high rain event, their salinities dropped down to single digits. But here at BB2, the salinities were still in the 20-30 range. So it's clearly not as well flushed as the other parts of Baffin Bay. So some conclusions about that. BB2 is not susceptible to flushing as the other areas of the bay. This supports brown tide abundance, and it's kind of a self-fulfilling loop in internal nutrient cycling, which then fuels more DON, which then fuels more brown tide. So we think that BB2 might be a seed stock of brown tide, and as it's flushed from the rest of the bay, it might slowly trickle out and start feeding brown tide blooms in the other areas, but it might take a very long time. We have not seen that happen yet, and it's 2018. So what does this mean for Baffin Bay? Still, nitrogen loading is crucial. High salinities, high DON, high brown tide abundances, and the long residence time means that it's very susceptible to nutrient loading, but it's also going to be very difficult to manage. And even though DON is high, just want to make sure I point this out, it's still fueling growth if you're putting more DO into the system. So any actions we take to reduce nitrogen, we can potentially see an effect. So in order to do that, though, we need to know where the nitrogen is coming from. Nitrogen sources to Baffin Bay are diverse, and they vary with rainfall. Here, Los Olmos Creek, which feeds into the BB2 site, is an intermittent stream, so it's not running for a large portion of the year, and there's no data really showing what's coming in through that stream. San Fernando Creek comes from the main city, Kingsville, and so that's mainly a wastewater discharge. And Petronila Creek feeds in from a large agricultural swath. You can kind of see here estimated nitrogen loads highest in the northern part of the Baffin Bay watershed. And also down here is showing different types of nitrogen sources. And you can see that agricultural and then atmospheric deposition are the main nitrogen sources. So Petronila Creek probably should be the place where we start if we're looking at doing some load reduction work. And that's exactly what we're doing. We started a stakeholder group with the goal of reducing nitrogen inputs into the system to decrease the severity and duration of algal blooms. And we've created a partnership with the local community members and organizations to achieve this goal. So we have the workholder group. We're still collecting data, and we're trying to come up with a protection plan, Texas State. And then in conjunction with that, we're also doing some restoration projects. And just recently, we found out that we're on the list of priorities for the 2019 Texas Coastal Master Plan. And we've already received funding to do some riparian buffer work in Petronila Creek. So this is the end goal, a healthy watershed. And I just wanted to go into some of the other aspects of this work group and what we're doing. We're conducting public meetings to educate everyone what's going on and get them involved. We've also worked with some local high school students that did some research projects in the Bay and then came to the university to talk about their work. So we're definitely working on engaging the public and making them interested and aware of what's going on. So I want to acknowledge all of our volunteers that help us out every month for 4 years, all the staff and all of the funding agencies. All right, and with that, I will take questions.
Any questions from the room?
What is the residence time or mean residence time of water in the Bay?
We are trying to calculate that again. The estimate we got was just over a year, but they didn't. The modeling they did to do it is not.
It's very rate related.
Yeah, it's all, and groundwater. We're still trying to figure all that out, but it's definitely very long. But the only estimates we have are rough. And just, I mean, usually for estuaries it's maybe weeks or, so over a year is definitely a very long time.
Most of those streams and rivers flowing into the Bay, are they kind of year-round streams? Or I used to work across Texas. A lot of people's good-sized rivers out there really only flow during the rainy season.
Can you repeat?
Oh. So the question was about how intermittent the streams were that feed into the Bay. The one that comes from Kingsville is year-round. It's mostly wastewater during the dry season. The other two are intermittent, but Petronila Creek, even when it dries up, there's still pockets of ponded water that can have their own algal blooms. And so then when it rains again, it just kind of pulses all of that stuff back out to Baffin Bay. But very, very dry overall. And then Los Olmos Creek is very intermittent. Very intermittent. Yeah.
It's showing up in Florida. Is it some of the same conditions that Baffin Bay?
So the question was about it showing up in Indian River Lagoon and if it's the same conditions as Baffin Bay. And generally, yes. The salinities are lower. It's still hypersaline, but not 70, maybe in the 40s in Florida. And it's definitely related to like sewage or DON there is what they're tracing it back to. So the question was relating Texas brown tide to another brown tide organism that's found like in Long Island, Aureoumbra. And I do not know. The question asked about why Aureoumbra was blooming in one area and then not another area in the same general region. And I don't know enough about that to be able to explain why those dynamics. But Aureoumbra, they are kind of put together a lot and talked about in the same manner in the way they bloom. Aureococcus tends to come in after other blooms have happened and crashed, and brown tide in Texas kind of might be the same way in this, the way that it feeds off the nutrients it does.
Any other questions? Okay, well, thank you so much.